Hello friends, our second topic is about the scientist. As we know about in our previous lecture is a general microbiology paint. In this lecture we saw about the scientist. Anton von Leeuwenhoek. It is a father of the microscopy. He invented the simple microscope which used only one lens. Second is a Louis Pasteur. The Louis Pasteur is a father of the microbiology. Is an important contribution in a germ theory of the disease. Okay, germ theory of the disease. Then the vaccines like a car vaccine. Here is a mnemonic for you. CAR. He invented the car vaccine like a cholera, anthrax and the rabies. Then the principle of the fermentation. So what he did is fermentation is used for the differentiation and identification. Fermentation means utilization of the carbohydrate, sugar, by the organism by absence of the O2. Okay. Acid can pH indicator involve, but few cases gas is a indicator. In case of the gas, we use the Derhan's tube. In the Derhan's tube, bubble is produced. Okay, and the acid organism which contain acid, it can identify the by the pH indicator. Okay, like a lactose fermenter in Mekong agar convert into the pink color, and not lactose fermenter can convert into the pale color. Same as a maltose is used for the differentiation of the meningococci and the gonococci. Maltose fermenter is a meningococci M for and the non-maltose fermenter is a gonococci. Okay, then the pasteurization technique. Then Louis Pasteur is in its name Pasteur. It's called the pasteurization method. Okay, it is a sterilization method of the milk. In these we use the two methods, Walder method and the flash method. Walder methods 63 Celsius for the 30 minutes, and the flash method 72 Celsius for the 20 seconds. As we know, as the temperature increase, time is a decrease. Okay, in older method 63 temperature, and the flash method 72 temperature for the 20 second, and the Holder's method for the 30 minute. Except the Coxiella burnetii, Holder method is not used. Okay, second is a water oven and the autoclave, also identified and by the Louis Pasteur. Water oven temperature is a 160 degree for the 2 hours, and the autoclave temperature is a 121 degree for the 50 minutes, and 50 pounds per square pressure. Okay, then the germline theory and the sterilization technique and liquid media. And liquid media we know as a broth. As you know, he saw the liquid media. This is a normal, this is a clear, and in this the turbidity is shown. So it is identified by the turbidity. Okay, the liquid culture media. So what is the Louis Pasteur do? So Louis Pasteur can do the germ theory of the disease, vaccines, car vaccines, cholera, anthrax, rabies, principle of the fermentation like lactose fermenter, lactose non-fermenter, maltose non-fermenter, pasteurization technique, Holder method, flash method, autoclave, water oven, and the sterilization technique. It is a part of the pasteurization, and the liquid media also identified by the Louis Pasteur. So it known as a father of the microscopy. Second is important is a Robert Koch. The Robert Koch, as its name, it induced the solid culture media. Okay, so what it is, the solid culture media, and the different organism produce different organi in a petri dish in a solid culture media. Then the bacterial isolation in a pure culture. Then the staining techniques. Okay, then the staining techniques also identified by these like a contrast and visibility of the organism for the staining technique. Dyes, basic dyes like aniline dyes and the it can identify by the these and the staining techniques. Then it can identify the cat. Okay, when we saw cat isolate of the organism like cat, cholera, anthrax and tuberculosis bacillus. Okay, then it gives the Koch postulate. What Koch postulate? It is a very important and you should know about the Koch postulate and which organism does not follow Koch postulate. So not follow the Koch postulate is a rickettsia, trypanosoma pallidum, mycobacterium leprae and the chlamydia. What should the Koch postulate is done? Okay, so in a first, microorganism is a constant associated with a disease. Here the should be associated is a very important. Then second is isolate the organism in a pure culture. First, the microorganism is a constant associate with the disease. Second, isolation from the pure culture. Then reinoculation. The reinoculation should produce the same lesion in experimental animal and they should be re-isolated from the lesion. Again, the re-isolate from these organism, experimental animal and presence of the specific antibody response. Okay, presence of the specific antibody demonstration, it is an immune response. So what is Koch postulate? The first of all, microorganism is a constant associate with the disease. That disease isolate the organism in a pure culture. Then these in a laboratory animal, these can produce the same disease. Then it can re-isolate from the experimental animal and the same organism in a pure culture. Then the immune response demonstrate of the antibody against the organism. Okay, so what is the molecular Koch postulate? The virulent gene present in organism is response for the disease. So it is a modified Koch postulate. Organism can't be cultivated in a vitro using the culture media like a microbacterium leprae, trypanosoma pallidum, chlamydia and the rickettsia. Okay, organism lacking the stable animal model is a Neisseria gonorrhea. Okay, then we saw the Edward Jenner. Edward Jenner is a father of the immunology. Then the Paul Ehrlich is a father of the chemotherapy. Is a Salvarson first antibiotic syphilis for the trypanosoma pallidum is made by this. Then the acid fast staining. Remember, acid fast staining is identification by the Paul Ehrlich, but modified acid fast staining or ZN staining by the Giles and Nihan stains. Okay, so it is a very important point to remember. Then side chain theory of the antibody and the standardization of the toxin and the antitoxin. Then the Joseph Lister is the father of the antiseptic surgery. 5% carbonic acid phenol is a surface disinfectant. Then the Caribimulis is the PCR and this is a not important to remember. Okay friends, if you like this also you can take screenshot, but this is not important for the inner you. Okay, so what we done, Anton van Leeuwenhoek, which is known as the father of the microbiology. Then we saw the Louis Pasteur. Louis Pasteur is a father of the microbiology. He identified the liquid culture, germ cell theory, then the vaccination like a car vaccine, cholera, anthrax, rabies, and the sterilization technique, pasteurization technique, Walter method, Fleisch method, and the autoclave and hot air oven. Then the fermentation is also lactose fermented, lactose non-fermented, maltose non-fermented. Then the germ theory of the disease and the liquid culture media. Then the Robert Koch is a solid culture media. Robert Koch identified the solid culture media. He isolate the organism is a cat organism like a cat, C, cholera, anthrax and the TB bacilli. Then the hanging drop method. Here we miss the hanging drop method. The hanging drop method is also identification by the Robert Koch. Hanging drop method is used for the appreciated motility of the organism. Okay, Then the staining method is also by the Koch, Robert Koch, and the Koch postulate is a very important. First of the then the organism is a constant association with the disease. The isolate the culture. Then this organism can same disease in a laboratory animal. Then the re-isolate the same organism in a pure culture and the demonstrate of the antibody against the organism. Molecular Koch postulate is a modified Koch postulate that tells about the virulence gene present in an organism. Virulence gene present in organism is responsible for the disease. Okay. Then the Paul Ehrlich is a father of the chemotherapy, and he saw the acid fast staining, not a modified acid fast staining. Then the standardization of the toxin and the antitoxin. Then the Joseph Lister is the father of the antiseptic theory. Then the Caribemulis is can identify the PCR and this and the scientist lecture. If you like, please share, subscribe my channel. Thank you, friends.